other factors that come into play, the orientation of the topography here and how it interacts with the current, all of the physical factors, oxygen and temperature and all of these other things, sedimentation rates, the type of, rock that we see down there. We just don't understand it, but we're trying. And so we'll see what happens a little bit later on in this cruise. We've got some shallower sites that are kind of in that real sweet zone that we're aware of. And so those could be really quite exciting. Have some high hopes. But this is part of science. We can't always pick the sweet spot.
No.
Sometimes we got to go around that sweet spot so we can understand it a little bit better.
That's the difference between science and documentary filmmaking, where you typically try to choose the best of the best of an area to present the most aesthetically beautiful and compelling picture of the place. Yeah, sometimes in science you've got to go to the bare patches, and that's part of the way you understand the whole.
Yeah. Well, I think Murphy's Law is alive and well out here, because as soon as we get on watch, the biology disappears, where I noticed that they had some corals and sponges and other things on the previous watch, at least the last hour that I was watching from my room.
Well, they are animals. Some of them are modal. They may have been running away from us. Chris.
Yeah, there's a few, but it kind of played out a little bit.
So Chris, one of our viewers wants to know if you were able to see a stylosterid in the earlier dive.
Yeah, that's a really good question. And I saw that they had zoomed in on these very small white colonies that was kind of on a little vertical face that they encountered. And I'm not positive. I don't know those that group all that well, but when I saw that, I was thinking stylosterid. The problem is that you could kind of see these little tiny, it was very small. You could see these little mouths and tiny tentacles around there. And it almost, to me, looked like they could be hard corals or scleractinians. Stylosterids are pretty hard too, because they've got but the question is, I don't know, but yeah, a good observation, whoever made that comment, and they very well could have been stylosterids. And I'm sure they're talking about the little white ones that we saw a little bit earlier that were tiny. They would have been impossible to collect because they were maybe only like an inch or so high, an inch or two high.
Do you want to explain for the audience, Chris, what a stylosterid is, since we don't have a visual example up here?
What, so you want me to exhibit my ignorance about this group? Okay.
Oh, come now. I know you know it all.
What's really kind of sad is that there's one that I've got my name on one of them too.
Oh,
from
wow.
Up here. So, and I don't understand the group all that well, but they're cnidarians. They're actually, if I'm not mistaken, in the larger group of hydrozoans. They're not necessarily octocorals or hexacorals. They typically are white, and they can often be very, very small, but there are some larger fan-shaped ones. They have an unusual sort of groove along their colony, I think in some places. They have a slightly different feeding style than some of the other types of cnidarians. They have a sort of a dense matrix, or sort of a harder matrix. When the polyps are sort of closed up, they tend to look like they have little balls on the end of the branches.
Oh, okay. Yeah, I've seen.
So, and that's almost about all I know about them. They're not that common in deep water. That's why we don't encounter that often, and that's why that was such a great comment from the viewer.
Yeah.
And it seems like it's Jason in Michigan. I don't know if that
well, way to go, Jason. Very good observation. I could watch, excellent, but I couldn't hear the comments of the people who are on that watch. So.
Yeah, we had so many comments, it was hard to remember. But yeah, thank you so much for that very extensive explanation of seamounts and the rocks to make it and volcanic activity. So I'm definitely learning a lot too. So what are some of the animal communities or animal species that we're expecting to see on this dive, Chris?
Well, just going on what I saw earlier, which unfortunately I don't see right now while we're up here, is there are some very large sponges. Well, not very large, but they were decent sized sponges. They were on a big thick stalk. And they had kind of this bulbous body on them that was sort of oriented sideways. And those were in the genus Calyptophora, which is in the family Rosellidae. It's a glass sponge, so it's got tiny microscopic parts that are made of silica as well. We've seen this guy before, and for the life of me, I can't remember whether we've ever collected one of these, so that is a possible collection target. That was the only type of sponge that I saw during the short period that I was watching after I woke up this morning. They got into some corals, and the corals that I did see were in the family Primnoidae, and there is no common name for the primnoids. It's just one of these very common deep sea families. And they got some beautiful close-ups of them to actually confirm that ID. And in fact, those could have been in the genus Neurella. What was a little unusual about them is they appeared very planar in structure. It was like a single fan of branches. A lot of times the Neurellas can be a little bit bushy, but there are some fan-shaped ones. But believe it or not, the way we often guess whether it's that genus or not is by the orientation of the polyps when they close up. You know, presumably due to the thruster blast from the ROV, or the water that comes through the thrusters of the ROV. And when they close, in Neurella typically, there are a few exceptions, but in that genus, when they close up, they orient downward. They're kind of like drooping.
Oh.
And there's another genus in the primnoids, it's called Calyptophora, and when they close up, many of those will close upward. I mean, they sort of close up their tentacles, and they don't completely retract into the branch. They just form kind of this little tiny column of plates. And so the Neurellas, the genus Neurella, will often close up facing down, and Calyptophora will often close up facing up. And so that's one of the clues that we look at to try to at least give a reasonable guess as to what it is. And I could see when I was watching that they were closing down.
Oh, okay.
So it's also, there's other little clues like the number of, they call them scales, the actual hard parts that protect the polyps that are called sclerites. And you can see the actual number of rows of scales along the polyp. That's another indication. Calyptophora has perhaps on the order usually of about 2 rows of these scales, where Neurella can have some more, and there's some other genera that can even have 4 or 5 or so longer polyps. So.
Oh, thank you. Well, it looks like it just made Jason's day. He really is excited about us answering his questions, and really.
Well, Jason, you just keep looking the way you're looking and observing the way you are, because that's fantastic. That's a great question.
Yeah, well, it's wonderful when our viewers are asking questions, because it gives us a lot more to talk about.
Yeah.
Yeah.
So Science Row, we're just kind of trying to make as much headway up towards this ridge as we can. If I did my math right, we probably only have about 30 minutes left in this dive, so if you have priorities, let us know.
The priority is to find a biologic or even image one, except for those little basket stars we've seen. It's been pretty barren here.
Put
so
on
I'm
the biological
watching.
radar.
I'm trying to find critters, but thank you, navigation. We've got our eyes hoping to see something.
Looks like a
Get our eyes open.
coral coming up in front of us. Oh, is that a sponge?
It's probably a dead stock over there.
Yeah.
That's why.
Yeah.
Oftentimes, if there's sort of a current pulse somewhere that's strong enough, you can actually knock the heads off of these stock corals. You can blow it off, and then the stock remains. It's actually glued onto rocks. This particular type of sponge that we've seen before, and so you just get this sort of broken up stock sticking up off of the rocks. I can't believe this. This is Murphy's Law here. Yeah. There's one little coral straight ahead. I think they've imaged that before, but if it's, I don't know, if it's the only game in town, we could take another look.
Chris, was that a coral you wanted to look at?
Yeah, I'll get the fancy machine here
Hey,
to
pilots.
circle it.
Chris is interested, I believe, in that coral right there.
It's just to look at a biologic one. Yeah, really hard to see. It's a little bit over here right now, I think. There.
Just that white spot on top of that sea star, is that what you're...
Yeah, something might be squatting on it.
Uh-huh.
A lot of these little brittle stars and other things like the squat on corals, they get off the bottom a little bit. They have a little more successful feeding if they can get off the substrate by a short distance.
Game down here, getting up in the water column. The higher the better.
Yeah. You know, maybe it's a good thing all the boxes are filled with rocks, given what we're seeing now. There's nothing to collect. We would have come up with empty boxes.
Yeah.
Okay. I think I already know what this coral is. But we can take a close-up. It's over here to the right, and they're sort of turning toward it right now. I'm going to take a wild guess from this distance, and I'm going to say that this is a it's got a new name, Remulogorgia militaris, or in that genus Remulogorgia. It used to be Pleurogorgia, which was a lot easier to pronounce.
And that's also an octocoral?
It's an octocoral.
Uh-huh.
And let's see if I guessed right. It's kind of a game that we play where we see if we can ID something from as far away as possible, because that's how you really learn. I got it right.
Here we go.
So this is Remulogorgia.
All right, we'll let you eat breakfast this morning then.
Yeah.
There's a very clear kind of nodal structure on there. Are those the polyps or the...
Yeah, those are the polyps. They look like they're closed up. I think when they're out, they're fairly sort of wide polyps. And it has kind of, you know, one of the clues you look out for this guy is it has kind of a blue-gray tinge to it, which is a little bit unusual. That's great, pilot. Thank you very much. It's kind of a common one, but we'll take what we can get at this point. Gallery has the old name. It doesn't have the new one because that's a pretty brand new change. Let's see if we can put a light on that. It's Ramula Gorgia.
And you said militaris?
I think so, but I think let's just leave it at the genus for now because there are others besides that species. It probably is that one because that's one that's known from around Hawaii.
kind of a flat, very flat ridge that we're on at the moment. You can see kind of a wide, it's definitely not very steep on the sides. It's kind of a plateau.
Chip just put in a little bit of a heading change, just FYI.
Yeah, right before we pull up, we might get into a little steeper terrain just looking at the map there, if I'm interpreting the direction we're going right, which I think I am. But it's going to be close. We'll probably find more animals on the steeper terrain.
You're probably going to start hitting this ridge now.
Oh,
Yeah.
great.
One thing that a lot of these corals, they often settle out on more consolidated substrate, like what we're seeing right now versus that cobbly substrate. And that's, corals, of course, don't have brains, but that's actually smart if they did have brains. Because if you settle out on too small of a little cobble or of a pebble and the current comes along.
Look, there's a sea cucumber just kind of winged by Argus.
There you go.
Oh, I see it. Was that a squid? It was hard to say.
Or a sea cucumber, I think that was a sea cucumber.
It
yeah,
looked
I think
like
sea cucumber, yeah.
Yeah, I didn't see it very well at all.
Similar to the one we saw yesterday, the purple.
Enypniastes?
Yes.
I love to say that.
How you say?
Yeah. Enypniastes.
Oh, that sounds like a Monty Python kind of designation. Enypniastes.
Eximia.
None shall pass. They do look kind of threatening, so.
Kind of a tentative AD because I really didn't see it. I saw it for about 1/2 second.
Yeah, I think it was that sea cucumber.
Yeah.
Enypniastes. Eximia.
Yep.
Yeah.
How do you know coral? How do you know
Bridge
corals
nav.
don't have brains, Chris?
No,
20 meters, 340, please.
Colonial organisms, probably. It's a distributed processing structure.
Oh, there's a...
Ooh.
Got another little
There's a
coral.
nice sea cucumber.
Oh, yeah.
Is that another one of the...
That's a beautiful deep purple.
Yeah, we could take a look at that.
Sounds good. We'll line up for a zoom.
I love how these sea cucumbers are translucent and you can actually see their internal organs and digestive tract through their skin.
Oh, there's also a coral right there, right above it too.
Yeah, that's another one of those, I think it's a Ramulagorgia. I cannot say that name. I could say Plurigorgia really easily.
Oh, I can try Ramulagorgia.
Okay, show me up there. Show me up there.
Wait, I'm really bad with names, but I think I got this one.
I can't hear you at all.
Well, that's a really nice still picture. Oh, yeah. Data logger, can we get a still picture of this? Well, of these two specimens, Ramulagorgia and
So I'm waiting for the zoom in, but I'm pretty sure I know the family. It's family Synallactidae. The pretty common deep sea sea cucumber family.
It's definitely, I'm getting audio, but
So
These animals, they cruise along the bottom and they eat a lot of the detritus. They're kind of the processing, the vacuum cleaners of the sea.
So our current name for this thing, we really don't know exactly what it is, but our guess, and this is from a lot of help from some sea cucumber experts, both in the United States and in Russia. Moscow's got a very well-known sea cucumber expert here, but it's in the genus Galathiopteria, I believe, and it's a question mark because that's what they think this particular sea cucumber is, but they're not positive.
it does. I can hear it.
Yeah, but that's our best guess right now. They're very distinctively brilliant purple, so you can see them far off. That Enypniastes that we saw yesterday was was really amazing because it was this brilliant ruby red.
Yes, these colors are just so vibrant, and I always wonder how that's possible to have such deep colors.
It's just kind of I sort of look at some of
I
these
don't think
things
they
that it
needed
really
the
doesn't
coral.
have that
They've
much
already ID'd
meaning
it.
because it's so dark.
Yeah.
There's no light down there. It's more just the way some of the the nature of their body reflects the artificial light from the ROV. You know, every animal's going to have a whole mess of different types of proteins and compounds, and some reflect life differently than others, but I don't know.
Yes.
Okay, it looks like we are getting in a little bit higher topography here. Yeah, so that I see the question from your audience about that coral. We think it was the same type of coral that we imaged just a few moments ago, that Remula-gorgia, and if that's correct, then no, it's not a rare type. It's actually fairly common at these depths. We've got a sponge, finally, a stock sponge. It's, I think, a little different species than the ones that they were imaging during their last watch. We'll take a quick look. Maybe not. Could be the same thing. So this is a glass sponge that we're going to zoom in on in a moment. And what that means is, again, that it has microscopic parts that give it structure that are made of silica. And that's why they're considered glass sponges. This particular sponge here is in the family Rosellidae, and specifically in a genus that's called Calyptophora. And it's got some very distinctive diagnostic features that I can see from here as well. First, it's a type of sponge that glues itself to the seafloor. There are other type of sponges that have these fine hair-like grapples, and they actually grab onto the seafloor, but this one tends to glue itself with sort of a plate. And then it has a stalk. Not every sponge has got a stalk, so that narrows down your possibilities there. And then one of the third characteristics is that where does that stalk insert onto the body of the sponge? And a lot of these sponges have sort of a concave surface and a convex surface, so a depressed surface or a rounded surface. And the Calyptophora, the stalk inserts onto the concave surface. And they were imaging that earlier in the last watch. You could see that very clearly. The stalk's going into sort of almost like a cavity. And then the sort of rounder face that we're seeing right here, even though it's kind of bumpy, it's kind of a rounder face.
Oh, that's a beautiful picture too.
Yeah. So do those folds provide more surface area for filtering out food from the water column?
Yeah, I guess so. I mean, that's a good guess. And it's not really clear to me which way they filter. The water could be coming in the backside and coming out the front side, or it could be other way around. It seems like coming in the backside would be more stable in a strong current.
Yeah.
Yep, so this is Calyptophora. I think trying to see how far we went on the idea of this sponge in years past. Let's see here. Okay, that's right. I was having a momentary early morning moment. This group of sponges actually has a genus and a subgenus name. So this is Calyptophora oxydiscus species. And part of the reason it has a subgenus name is because they're not very well known, and then there needs to be a lot more work on these to really separate them out properly. thank you very much for the imagery pilot.
Chris, sorry, are you trying to talk to us up here?
I was just thanking you for the close-up.
Oh, okay, very good.
You can tell us to shut up if you guys need to talk and work something out, or just
doing some training.
We got a good mute button, don't worry.
Oh, that's good.
Just
Because I
shout
mean,
if
I've
we don't
had
hear.
2 big cups of coffee, so I'm a bit chatty right now. Oh, could we image this particular coral, please, if it's possible?
This particular coral.
Yeah, this particular one. I think they imaged this before, and I think this is a bamboo coral.
That one is a little one, right?
Yeah, it's a very little one.
We saw bigger ones in the last watch,
right.
the last dive.
Right, this might be a younger version of that same type of thing that we saw last dive, which were quite long.
Yeah. Can we tell how old this bamboo coral is?
They could with this type of coral by looking at the hard part. It's got sort of a hard matrix. And that's kind of clear of polyps right at the base of it, if you take a look. But I wouldn't be able to tell you how old this thing is. I don't think it's particularly old, given it's small.
Yeah.
Yeah, it looks like it's very similar to the one they imaged before.
Mm-hmm.
And
the polyps look very, very young and rosy.
Yeah, so polyps on these bamboo corals, which is they just did the revision of the taxonomy of these, and it used to be in the family Icididae, and now it's in the Keratoisididae. The polyps tend to be large, and the oral part around the tentacles in the mouth are often kind of pink or red, and that's another kind of diagnostic versus some of these other unbranched corals. I wonder if you could zoom down by the base just a hair. And you see that little black band around
yeah.
it? So that's why they're called bamboo corals, because they have these occasional what they call nodes. And that's where the skeleton is not calcified. It's actually proteinaceous there, and the protein's called gorgonin, and it's exposed. And it shows you that underneath all that white calcium carbonate is the sort of black gorgonin that runs up through the whole stalk. But that particular part is bare, and it's not because an animal did that. That's just part of it's the way it works.
Oh, and this one only has one.
Yeah, it just
the other ones that we saw that were about 3 meters long,
right,
had quite a few.
Yeah, one of the ways they're looking at trying to figure out this whole group is what is the interval between the nodes. That's one character that's important. But mostly they look at the microscopic parts in the polyps, which can also tell them a lot, and we can't really see them right now. But, you know, whether it has branches, it doesn't have branches, whether it has a node, doesn't, you know, how many nodes, how they're spaced apart.
And those nodes is also made of keratin? Is it like
it's gorgonin, I believe, and Steve knows these. Steve Oskowitz, who was on earlier, knows these better than I do. But I'm pretty sure the protein is called gorgonin.
Okay.
Thank you very much for that close-up.
And the viewer here says that in the last watch there was a conversation about whether sponges are single organisms or colonial. Could you clarify?
So they're not a colonial organism. They're a single organism. They're very, very simple. They're filter feeding. I'm trying to think of what else. So corals can form colonial organisms where you have a number, have a number of sort of, you know, the polyps themselves, they can be all connected and attached, but it's not quite the same with sponges.
And they don't have stinging cells like corals do.
Right. I see that Jason asked another question about Caulophacus versus Bolosoma, And the difference, very obvious difference, Jason, is that where the stalk actually inserts onto the body of the sponge, in Calyptophora, they tend to insert, the stalks insert on the concave side of the sponge. And in Bolosoma, the stalk inserts on the convex side of the sponge. And at some point we'll come across one of those and you'll see it. It's quite clear distinction. So that's one way that we can tell them apart. The genus Bolosoma is in the family Euplectellidae, and the genus Calyptophora is in the family Rosellidae. So good question.
Do you want to do one more ship move? We're getting up on the end.
Got any more questions?
Yeah.
Somebody said sea cucumbers don't have brains, and that's correct. They don't. They have a very simple nervous system. They need nerves in order to contract their muscles, particularly the swimming ones, in order to be able to swim, but they don't really have brains. Looks from,
can you see this?
yeah, I'm looking over your shoulder if you don't mind. I'm sort of peeking. Looks like Frank from Liverpool, UK, he was asking whether or not we have any UV lights to show any luminescence on the seafloor. No, I'm afraid we don't, Frank. There are some fluorescent animals down here that if you touch them, they'll fluoresce. But again, because we're in a monument and trying to preserve animals, we're not going to go around squeezing them.
Thank you.
No poking and squeezing. We like to do that on land.
Unlike the rocks, we do squeeze the rocks
right.
to see if they're weathered or not, and what kind of rock would suit either the microbiologist or the geologist. There's two different preferences. The microbiologist likes the soft, squishy rocks, and the geologist for dating likes the hard, angular rocks.
You know, I've never heard of a rock being described as squishy until this cruise. And Dr. Orcutt described it. And you know exactly what she means. I just never thought of a rock being squishy before.
Yeah.
Yeah, I'm learning a lot about rocks here. Rocks tell a story.
Yep.
Yeah, just to get back to... Oh, sorry.
No, I just wanted to say that goes back to our geologist. She dates rocks. There's a love story in the rocks. So that really grabbed my attention. And now I'm just learning so much about rocks. Pillow rocks and fluffy rocks.
Well, rocks are fascinating. They really. I grew up in a family of geologists. I'm the black sheep here.
Yes, I remember you said your first toy was a hammer, or what was it?
Yeah, before I had a dog or a pet or anything, I had a pick in my hand, and my father was dragging my brother and I up into the mountains to collect minerals and things.
Oh, that's amazing.
It's a fun way to grow up, frankly.
So you really did have a pet rock first.
You know, believe it or not, I did have a pet rock.
I believe it.
Whoever thought of that, I mean, wound up being probably a millionaire, just taking a rock and putting it in a colorful little box. And selling
All
them.
about the packaging.
Yeah, I know.
Presentation.
Yeah. Well, I'm very interested in the microbes on the surface of those rocks that we're collecting.
Isn't that a bit small-minded of you?
No, I think it's huge. No, I mean, I'm really interested in those microbes because I believe they really form the foundation of the food chain.
I know, you missed the joke. Small-minded, little microscopic arguments there.
Yeah,
That's
no, I kind of got it. No, I was reading a couple of messages there.
No, they are so important. That's why we're delighted to have Dr. Orcutt out with us on this cruise, because oftentimes we don't really investigate that part of the environment here on these dives. You can't.
Yes.
Yes, the microbes are often overlooked.
Yeah.
As Chris mentioned earlier, we are very careful about the collections that we can make because this is a fully protected area, and as part of our permit, we only have a limited number of things that we can collect, and basically those protections are put in place to preserve this place for future generations. And actually now NOAA is considering a sanctuary, National Marine Sanctuary designation for this area. And there's a public comment period for that just opened yesterday. So that would layer on additional and more permanent protections for this entire section of the ocean in the marine realm.
And where can people find more information about that?
Yeah, so if you go to our website, papahanaumokuakea.gov, that's papahanaumokuakea.gov. You can find information about the sanctuary process and the upcoming meetings. They'll be virtual, of course. So in December, we have 4 meetings. And also, but you can provide comment online in both English and Alelo Hawaii, which is the Hawaiian language. So if you want to participate in that, please go to the website and take a look, and we would love to hear from you. Chris is probably has been studying the deep sea environment in this area for many decades, and through his work previously with University of Hawaii, and he had a Hawaii Undersea Research Laboratory at the university, and came up here. When was the first time you came up here, Chris? It was quite a while ago, huh?
You know, I was trying to think of that. I think it was 2001 was the first time I came up. And then the university was also had a really old, relatively shallow submersible called the Makali'i submersible. Could only go to 1,200 feet, or about 400 meters. And the very well-known Dr. Ricky Grigg was coming up here around French Frigate Shoals. He was a coral biologist, and he was one of the first, I think, who's investigated. This is before a monument, before a reserve or anything.
The wild days.
Yeah, and he came up here and kind of got the ball rolling a little bit and trying to understand what's up here.
Yeah, and that was what kicked off some of the initial protections for the marine area.
Yeah, could we zoom in on that anemone as any chance?
Anemone.
See if you can.
Sorry, pause for
That's
biology.
at the end of our leash, but we can make our way over there, and the ship is still, Argus is still moving, so we'll get there in a minute. So we're going to, I don't know if you caught that, we're going to go to that for that anemone.
Well, you know what we used to say in Hawaii, if can, if no can, no can.
So the ship's finishing up a move. Argus has to swing in a little bit, but with a 3-hour rise time, it's time to go, unless the chief scientist wants to change anything about the dive.
Well, if there was a few more critters on the seafloor, I might be tempted to extend it, but I'm not really being treated very well as a biologist by this part of the seamount. So I think we could probably recover in a bit.
Okay.
But let's get a look at this anemone. I think I know what it is already, at least the family, but it'd be nice to confirm that. My guess at this distance is in the family Hormathiidae. It's a fairly common deep sea anemone. And when they zoom in on it, you'll see that the stalk has got these, it's kind of rough textured with little tubercles on it. And, you know, but we'll find out as soon as they zoom in.
Oh, yeah, looks like you were right.
Yep, all those little bumps. Hormathiids tend to have a very kind of, I don't know if it's chitin or what, but they have this very rough textured stalk. Right underneath the row of tentacles there, there'll be kind of a little clear zone, and then they have sort of almost like a double layer of tentacles that are very small compared to other things. And so that's kind of a classic Hormathiid. And I'm going to see if in the gallery we've gone any further on something like this. Give me a moment to check my cheat sheet.
That stalk reminds me of like the like papaya, like the stem of a papaya. Looks like a lot like that, right?
Oh.
Well, you know, on some of them at least, we've gone to the genus Philiactis. It's not a.
This one.
Yeah, see, well, it's a little different than this one. You know, I would say we could put this as Philiactis species question mark on there. And some of these names, we don't make up these names ourselves. We get a lot of help from taxonomists all over the world. With this particular group of anemones, it's Estefania Rodriguez in New York, who's looked at a lot of our pictures and really helped us out on anemones. Yeah, I think that would be the best ID. The depth range looks pretty good too.
And what's the depth range right now?
Right now the depth, what is the depth we're at right
now? 2756.
Okay.
Oh, I just know, as you said, volts are too low.
So Chris, how do they were able to contract those tentacles quite quick? Is there some process of like pump out liquid?
All right, we're setting up for recovery up here.
You know what the mechanism is for how they're able to collapse upon themselves so quickly?
I don't know, but I suspect it's more muscular than it is sort of a water pumping thing, because they're pretty rigid, tough little anemone. So
fleshy and tough. It's not just like a bag of water.
Okay, are we lifting off right now?
Sorry, I don't know if you heard that, Chris. Yes, we were setting up for recovery. You're not on SPL, Trevor.
I'm not on SPL.
Okay.
Now I'm on SPL.
So we're going to have a moment of silence here to do sort of a little ceremony here.
Great, yeah.
And we are doing this protocol to acknowledge and honor the biocultural resources that are present and that we are interacting with and impacting. So we will be back on SPL shortly. Thank you. So we'll go all on mute and okay, so we are starting to bring the rovers up to the surface. And how long would this process take?
It's going to take a while.
A couple of hours?
3 amazing hours.
Oh, 3 amazing hours. Wow.
Ish.
Okay. But as we're coming up, we're also surveying the marine life, right? Could we collect anything on our way up?
I don't think so.
No?
I don't think they stop on the way up.
Oh, okay.
Got places to be.
Wow, 3 hours.
Yeah, there's no Niskins or anything on the way up, right?
What I could see from Meredith, she was saying, can we take a Niskin before we leave the seafloor? I just saw her in the chat.
Sure, roger.
I should be looking at the chat more often. I apologize.
Aaron, video, please, full super extra wide on Herc Zeus. Science Row, can you please confirm which Niskins I have available? Or which Niskin you want this one to be taken with?
Yes, we have Niskin 3 available.
3, sounds good. Oops, that's not going to work.
So the Niskins are essentially water samples. It's a little device that allows us to collect some seawater at the location that we're at.
They're kind of like a big sort of hiking water mug or something with a top. And they pull the little string to release and open up the top to allow the water to go inside. And then it closes very quickly and seals it off because you don't want the water mixing.
Yeah.
Collect with.
And are they going to use that for eDNA analysis?
Yeah, that's good. And Andy, by now you should know what that is. Maybe you could explain. You could explain it.
So as part of the revolution of being able to categorize DNA in situ or in the environment.
Let's get out of here.
There's a process called using environmental DNA. And basically what that is they take a water sample and they bring it back to the lab, and then they they're able to extract all the DNA sequences that are in that water, which are representative of the animals and other critters in that area. If you have, some of these animals have mucous membranes and other things that shed biological material in the water column.
You can kill your auto heading.
And those, if you break those out in an eDNA sample, you could potentially identify the organisms that are in a given area by that biological material that they're continuously shedding. And
science, I'm sorry, if I can interrupt before I forget,
Yes,
can I get the sample number on that Niskin?
Yes, the sample number for that one was NA134 024.
I just figured that out now.
Roger, thank you.
Yeah.
Yeah, and so they're developing
12 right now?
Database of organisms based upon their genetic structure. And so from that database, if they pull out a sequence of DNA, they compare it against that database and see if they can identify those organisms in that area by that sample. How'd I do, Chris?
You did pretty good, I think, but Meredith is your judge.
Ah.
So if she comments on your description, we'll see in the chat.
Okay, give me a grade, Meredith.
She said you did great.
Oh, yeah. Gold star.
Thank you, Andy. I also learned a lot from you.
She's given you an A. She didn't give you an A+, but she gave you an A.
I'll take it. I'll take it. It's 4:30 in the morning. It's excuse me. It's a little later. Not my prime operating time for my brain yet. But yeah, we're using that also in shallower waters and coral reef environment, collecting samples for eDNA analysis and the reefs on the reefs up above. And
there, 15 meters a
as
minute.
many things in science, this takes a long process of accumulating and building out that database before it really becomes fully useful, because certainly some of those organisms, their DNA has not been sequenced. So if they're not in the database, then it might not show up when we analyze the DNA sample. But over time, that database will expand and become more and more useful for these eDNA analyses. If you're a diver in a shallow environment and observing corals and fish, you don't always see everything. There are a lot of what we call cryptic organisms that hide in the rocks and aren't easily seen during like a visual survey, but they may show up in a eDNA analysis. So that potentially will paint a much more complete picture of what's in a given area. It's very exciting, new technology.
Do you see Meredith's comment in the chat right now?
No, I wasn't looking at the chat.
Let's see it.
Yeah, she says that when we see a new sequence in our eDNA, we flag it, assign it to a family, and put it in a note that future work should look for new species. put in a note that future work should look for new species in that area. Yeah, thank you, Meredith. I wasn't following when we went down. Did we, on our descent, did we see anything interesting cruising in the water column?
I didn't see the ascent, but the biologist abuse continues. The water looks pretty barren right now.
It does. Well, there's some things.
Biologist abuse, I like that.
Every once in a while it's like that, and then sometimes we'll get into one of these communities and the geologists get abused while the biologists go crazy with all the animals. So I guess all's fair.
All's fair. Love and research, all is fair. So where's our next dive site, Chris?
It's going to be on the same geo, but it's going to be on the north side. We're on the south side right now,
okay.
And it's going to be a long one. And and it is going to be more of a biology dive. We're going to change the jaws on the manipulators so we're able to more easily sample some corals without destroying them. We could snip little branches really efficiently.
Nice. Little underwater scissors. Yeah,
we're looking at potentially a 24-hour dive tomorrow. So, and we only have a short transit.
Yeah.
That could be really wild. I'm not sure everybody's going to stay with us for 24 hours.
Chris,
the hardcore will.
It's just on the other side of this seamount?
Yes,
okay.
Exactly.
Thanks.
I think Emil told me it was about an hour and a half transit, so it's going to be pretty quick.
Okay.
Yeah, and as you've heard us say before, orientation to current is the name of the game for a lot of these organisms to provide as much opportunity to capture things, filter things out of the water. So when we're looking at the seamounts and looking at different ridges in different orientations around the seamount, they will have different communities because some of those ridges are more exposed to stronger currents and therefore potentially better for these filter feeding organisms. And so we could see very different communities on even a smaller seamount with different ridges extending in different geographic directions. And sometimes the currents are too strong and they actually can't attach to those ridges or that substrate, even if it's potentially good habitat. So that's the other consideration for these organisms. But it's all about the current and orientation to the current, getting up into the water column to have more exposure to those currents and the critters that are floating in the current that they collect for food. So you can really observe that in a lot of these environments. And I love watching how they attach behind rocks, just like in a river as the current's flowing down. Behind rocks, you get these eddies where the water goes over the top and then circles around. So a lot of these organisms, they attach right behind a rock, not on the top of it, and it gives them multiple opportunities for collecting food out of the water column because the currents are eddying, going around in circles in that area. It's really interesting watching where they attach and what the dynamics might be going on that drove that particular attachment site.
So just so you know, Dr. Asako Matsumoto from the Chiba Institute in Japan, she has joined us at the end. She's been very, very busy, but I think she'll tune in when she can tomorrow. And she's a coral specialist, one of the world's premier coral specialists, and she's particularly in a group called the Plexauras, which Steve Oskovich is also very interested in. So I'm hoping we'll get up in the depths where we see some of those animals. They're a very complex group, just like Steve mentioned in his talk, like that. And it's great to have a few people really working on this, trying to untangle the taxonomy.
Those were the corals that were making him question his life choices as
yes, that's exactly
for the complexity of the taxonomy. And
well, you gotta have some courage to work on those, because they're hard. They're harder than a lot of them to really figure out.
Yeah, you don't realize how dynamic a field taxonomy really is until you get on a ship with a bunch of taxonomists, and the discussions just quickly degrade into esoteric from the outsider's perspective, mumbo jumbo. But it's very interesting the way they classify these organisms and move families around, and genus in particular. And it's this is from often when we collect an organism and we say, wow, we have never seen that before. You know, it may be a new species. We definitely don't say it is a new species, because the process to actually identify and categorize that animal can take years. The sample may be sent around the world multiple times to different specialists to try to identify it. So it takes a long time to actually determine if it is a new species or if it has been found, because these experts are spread across the planet, and it takes quite a while.
Thank you.
The wonderful sort of new thing that's being applied to both deep sea corals and to deep sea sponges is the field of genetics, which is, you know, Meredith's field and some others, because particularly with these groups that are just so hard to identify morphologically, at least in situ, you know, you have a better chance if you can collect it and look at them under a microscope. Yeah. But still, they're very, very difficult, whereas genetics is such a precise field of study that we've got a chance. The corals were a little bit easier, I think, to do genetics on than sponges, but there are some people who are making some progress in sponge genetics as well. And so I imagine the next decade or so, it's going to be extremely interesting as the whole field of deep sea taxonomy evolves more to a genetic-based taxonomy versus a morphological-based taxonomy. Not to say that morphology is, you know, gone or out the window. It's always going to be there. But now we've got this other tool that we can marry with the morphology and really start to understand, you know, what's going on, where these animals really belong, what group they belong to. Because a lot of times things can look the same and they're really not. They're quite different. Yeah. So when into the room here, one of these guys who's still questioning his life choices, Steve Oskovich, just came in. And we were just talking about plexaurids because Asako just got on the chat. So you don't have to worry about biologics. Not a single biologic was collected, and all the boxes are filled with rocks. Yeah, that's true. Yeah, we'll have to look at the pickings on the rocks. This looks like leftover Halloween candy, isn't it?
Yeah. Yeah, it's waking me up. I got these Smarties.
It is leftover Halloween candy, yes.
Thank you.
Yeah, thank you. I see another message from Meredith. She was saying, and she knows this, of course, being a geneticist, that sponges can be tricky. They have a lot of inhibitors and not a lot of tissue, but she is also getting DNA out of them in their lab pretty consistently now, which is wonderful news. I know it used to be a problem probably a decade ago or so trying to do this, and they're getting success now. They're figuring out techniques to actually do this.